Hello everyone, once again. My name is Phytoremediation. I belong to Department of Plant Biosciences, and I'm also a member of Agri Innovation Center. This is the second video to introduce the researcher in our laboratory. And in this video, I'm going to talk about identification of novel cesium-specific transporters and their potential use in phytoremediation. I think many of you know that only a very few percentage of land has been used for agriculture, that is around 37%. And among these 37%, only 23% is being used for crop production. And by 2050, the world's population will increase by about 34% to 9.1 billion, but the potential cultivable area will increase by only about 5%. And soil pollution is a major threat to food security. Currently, 33% of the Earth's soil is contaminated. So you can understand from this slide that we have to feed more people using limited land, and soil pollution adds more complexity to this issue. So it is important to develop technologies that can clean up soil and make more lands available for agriculture. So in the next slide, I'm showing you what is the condition of soil pollution. And as you can see, it is around the world. And mostly these are heavy metal contamination and also some toxic metal contamination. And Japan is not different. Here I'm showing three major contaminants: arsenic, cadmium, and cesium. And you can see it is pretty much all over the contamination level. And among these, cesium became very important, one of the important contaminant these days because of this Fukushima disaster, as you all know. And from this nuclear disaster, actually, some isotopes of cesium has been delivered to the environment. And these are basically, we can divide cesium in three isotopes. One is 133 cesium, that is stable isotope. It doesn't do any harm. Other two are 134 and 137. These are radioactive isotope, and their half-life is like 137 cesium is 30 years, so it's very long, and this is very toxic for human. And obviously, as you can see, it happened to the Chernobyl. That same thing happened to Japan too, and you can see here very clearly that this area is highly contaminated by cesium. So now, the concept-wise, you have contaminated soil, and now the thing is whether we can we clean up the soil by using the plant. Yes, this is a technique we call phytoremediation, where plants are being used to clean up the soil. For example, in case of cesium, this is a cesium contaminated soil, and we have in the cell there are some cesium transporters, and that will take up the cesium inside the cell, inside the plant cell. But if it is uptaken at a limited number, then you will not be able to clean up the soil very well. But if we can increase these transporters, then we can incorporate more cesium in the plant, and then this will be like this. We can clean up the soil. So this is the basic of phytoremediation, basic concept of phytoremediation, and this is possible. But the problem with cesium is that it is very closely associated with plant nutrient potassium. Potassium is very important for plant growth, and many cesium is being uptaken by potassium. Okay, now the question is, if we want to use the phytoremediation technique and we decide to overexpress potassium transporters, then what will happen? Look at this slide. This slide we show that if we overexpress the potassium, then potassium transporters, then what will happen that it will transport both potassium and cesium, and it will make the potassium depleted soil, and this will be unsuitable for cultivation for future. So based on that, we thought that, okay, whether we can find any transporter that is potassium independent, but only take up cesium. So the concept is like that. In that case, the potassium will be retained in the soil, and this will be suitable for cultivation for agriculture. So then the next question comes that, are there any potassium independent cesium transporters? And if there are, how can we find it? So we went back to a very old literature, and we found that the beautiful work done by Hampton et al., where they showed that some of the ABC transporters are only being specifically altered by cesium, and they are not affected by potassium. So we thought that this could be one potential candidate. And I will show you ABC transporters have been used for across the species from human to prokaryotes to transport different kind of substrates, and this does not have any substrate specificity. And particularly in plants, you can see that it has a huge number of ABC transporters, like 130 in Arabidopsis and 133 in rice. So that means these are really, really important for plant. So obviously we focused on this, and first thing we wanted to do is that, because as I showed you in the Arabidopsis, there are 130 ABC transporters. So we collected 80 mutants of these ABC transporters, and we wanted to screen them against cesium. So for that, we initially need to develop a cesium screening method, and we did it here. You can see here with 1.5 millimolar cesium. After day 3, the roots grow only 50%, and the leaves are bleached too. They become white. So this is a very nice screening system and visual screening system where you can clearly screen the mutants for their resistance to cesium. And based on that, and after we did 80 mutant screening, and we found that one of the mutants called ABCG37, double mutant called ABCG37, ABCG33, double mutant is highly resistant to cesium. Okay, you can see here this is becomes white, the leaves becomes white, but here they are still green, and the roots are growing very well. Okay, so based on that, we thought that this ABCG33 and ABCG37 may be cesium-specific transporters. So to prove that it doesn't, as I told you earlier, that cesium is involved with this potassium, and to confirm that these proteins do not uptake potassium, so we use different, we measured the cesium content in different concentration of potassium, and you can see here we found that irrespective of the potassium content, always we saw there is a decrease in cesium content. So that means these potassium, these proteins probably do not have any effect on potassium uptake. Now, we also look at the growth phenotype of the plants. As I told you, that potassium is very important for plants to grow, and you can see here with enough potassium, the plants grow very well, and less potassium, plants grow very less. And if these ABCG33 and 37 proteins uptake potassium in this mutant, because they're absent, the potassium uptake will be much less, and they will not be able to grow. They will be very, very sensitive to potassium depletion, but in reality, they grow very well. So that means they're independent of potassium. The next thing that we did is that we wanted to know whether really they have any transporter activity, and we expressed them in yeast. And in the yeast, you can see here that yeast expressing ABCG33 and 37, irrespective of the whether it's a low concentration of cesium or high concentration of cesium, always we see more accumulation of cesium inside yeast, the transformed yeast. So that means these are really cesium uptake carrier. And then we also confirmed with the yeast that whether it has anything to do with the potassium, and you can see here, you can see here, this is a yeast strain that doesn't have any potassium carrier, and we express ABCG33 and ABCG37. The idea is that if they uptake potassium, then in the potassium depletion media they will be able to grow, but they did not grow. So that means these proteins do not uptake potassium at all. So if I summarize, then for the first time in the world, we identified plant proteins that transport cesium independent of potassium. And these plant proteins can be used to develop plants to clean up the cesium contaminated soil. And thank you very much for listening. And if you're interested, you can also look at our lab URL. Here is the lab webpage. You can go and visit our lab. I would like to thanks once again for your patience. Thank you.